before we get into the show, we figure we drop a couple little bits of announcements for you. Imminently dropping is going to be the Hazard Class podcast with our own Jake.
Yes, he's doing a phenomenal job. He's very entertaining, very funny. He's got a great sense of interviewing, great sense of being able to pull information from somebody. I can't recommend this show enough. It's going to be a hit.
Check into your iTunes, Spotify, wherever the hell you get your podcast from, where we get this drivel from. Go and look for it. I know it's going to be dropping in there in the next week or two, and we will let you know on social media. It'll be a huge blitz when it happens, but huge show. And we got a whole bunch more showing popping up soon, so keep your eye out for that stuff.
And don't feel like you're cheating on us. We recommend going and watching these shows. Yeah, absolutely. But if you do want to show support to us, you can always do that by the THMG and the Hazmat AF swag that is on the website. So go there and buy some really kick-ass T-shirts and support the show. If you wanted to support the show in other ways, you could always buy Bobby a drink. Where could they find you, Bobby?
Hot Zone, October 17th. We'll be out there doing some stuff, and then CBR and E Convergence in Orlando, which is October 20th. So let's just get into the show. Hey, everybody. Welcome to another episode of the Hazmat Guys.
We are here, and we got an exciting day because we have not scared off a previous interviewee, and she's decided to come back on and share more of her experience with us.
We have Jacqui Hardt here you might remember her from way back in episode 136.
Yep.
Unbelievable. And she is our bio person.
Sure am. I am fantastic.
Unbelievable. We actually tripped across each other in Phoenix recently at a conference put on by Label Master, and it was just like a catch up, and it was great. And we started talking, and then she wrote this whole outline from the hot tub, and it was exquisite.
Best place to come up with an outline, I tell you.
Couple words. I would try it, but I promise you I'll drop my laptop in the water with me, and it's
like a toaster in there.
And then without a note, nobody knows what happened. And
so, all right, it's been a while. Episode 136 is back 300 episodes ago, so it's probably a couple of years ago.
I think it was 2018, maybe. It was way back. 2019.
An update. What are you doing? Yeah. Update us. What are you doing? Where are you now? What's happening?
Well, actually, I haven't gone very far at all. I'm still in exactly the same place I was when I recorded 136 with you. I'm still based in San Diego. Go Padres. Doing good. The postseason was there last night. So if my voice is a little rough, that's why.
And just to be clear, you had nothing to do with COVID, right?
It was not my fault.
Okay,
but
just want to make sure.
It kept me busy. Biosafety professional in a pandemic is a slightly important role, and I have never worked that hard in my life because everybody in the world was trying to avoid getting COVID, and I was trying to protect the people who were playing with COVID in labs from getting COVID and spreading it to everybody else. So yeah, that was fun.
Well, oh my God, that's how I would have never. It's funny how you do something and then the world around you changes and your role in what you're doing completely changes to fit the change in the world around. That's
unbelievable. All right, so one of the things we're going to go through is the classification of bioorganisms, and I know there's like 1 through 4.
Yes.
Okay.
You have remembered correctly. So microorganisms are generally bucketed into 4 risk groups. Risk group 1 is the lowest. Risk group 4 is the highest. And depending on their potential to cause serious disease or even fatal disease and their ability to spread, depending on their route of exposure, that buckets them into these different categories. Some common examples of biosafety level 1, risk group 1 organisms are things like yeasts, E. coli that doesn't cause disease, really anything that is unlikely to cause a disease in a healthy person. They are your general, can be used in a general basic lab environment with very little biosafety controls.
Okay, because I was going to say like yeast, like you're talking about what I would make bread at home with.
Like yogurt.
Yeah.
Yeah, because my wife's going to be really annoyed if I'm in a full like level A suit making sourdough.
No, you're good.
Okay.
You can still take your probiotic, no problem.
Yay.
And once it's out of risk group, we are talking into risk group 2, we're talking pathogens.
Okay.
And so there are many, many operations that deal with risk group 2 pathogens at what is a control level or biosafety level 2. And these include, the things that will make you sick, but probably won't cause serious disease or fatal disease, and are not usually air communicable. So we're talking, strep, we're talking some of the hepatitis. We're usually looking at less concentrated clinical samples. We're not really growing things in highly concentrated format. I mean, some things might be, but most of the time it's pathogens that are going to make you ill, but not really, really ill. And they're not going to communicably spread to a very wide population or escape the lab and cause a large illness event with people outside of your space. And also they often have vaccine or prophylactic treatment or clinical treatment if you are exposed and contract the disease that can limit or eliminate the symptoms. So very common.
So what would a BSL-2 lab look like? Like if I were to envision it as a first responder, like I'm going in, what things would I expect to see and find and come across?
It actually pretty looks very similar to a standard lab, like a chemistry lab or a biosafety level 1 lab. But there will be a few indicators that you see as you enter. They may be biohazard symbols on the door. They will be probably labeled BSL-2.
Okay.
And when you go inside, you will start to see things that are labeled with biohazard symbols, fridges, freezers, containers. Also, there will likely be biohazardous waste in there, which is in big cans, often red or yellow or orange. Depends on the state you're in. But they have giant biohazard symbols on them. And there will likely be a bit more control relating to personal protective equipment and access and just general items to stop spread of the pathogen they're working with.
So you're going to have gloves, masks.
Yeah.
Are the
maybe not masks or booty. It depends on the science. When you get into biosafety level 3 is where you're really dealing with respiratory protection. So biosafety level 2, risk group 2 materials are often not air communicable. So the need for respiratory protection, especially if you're working within a biological safety cabinet, which is the primary engineering control for handling pathogenic materials, which you will very likely see in a biosafety level 2 lab. That's the little cabinets they work in, which A, keeps their material sterile, but also it stops you from inhaling or from splashes of that pathogenic material when it's being manipulated. So it's standard lab PPE generally. It's lab coat, gloves, safety glasses.
The animals that are involved in the biosafety lab too, right? I think lab, and quite often I think just like a rat or a rodent or something like that. But they are all different types of organisms. Are they kept in the actual lab, or are they kept separate? And is that still considered part of the lab where their cages are?
Animal facilities are usually completely separate entities. So what I've explained for BSL-2 is kind of your standard lab for playing with petri dishes or samples or growing cultures in a microbiology setting.
Okay.
When you get into animal facilities or insect facilities or plant facilities, you usually have an entirely separate portion of the building or an entirely different site in certain circumstances, because the laboratories for housing live organisms are inherently more complicated because they're live organisms. You are trying to not only do science and research, but you're also trying to house and keep specimens alive to A, conduct that research, but B, there are a lot of ethical requirements for doing live organism research that have to be met, just because that's the rules for when you work and perform research with live animals.
So very similar to being a parent.
Well, yeah, the containment might be similar, all those bugs and germs. But yeah, it's essentially, you want to maintain the integrity of your research because they are conducted under very strict parameters with live animals.
Okay.
Because you have control groups, you have non-control groups, you have to control the parameters of how that experiment is conducted. But also, you don't want them to escape. You don't want them to become contaminated and influenced with something brought into the facility that could impact the viability of either the organism or the study. And often, these organisms are highly specialized, specially bred. Some of them are genetically modified, so they have a certain disease trait or they're immunocompromised. So they are extremely valuable specimens to the researchers. And if you are starting to do studies on live animals with, say, a drug product or a biological or a pathogen, your project is far along in its life cycle. And you are at a critical stage where that data could determine if your product is launched or if your drug is approved by the FDA or whatever the science you're doing. If that animal study fails, that could be to your project. So you have so many different parameters. So that's why these facilities are often very specialized, very isolated, very secure, because also this kind of research is not always socially accepted. We have a lot of people who may not philosophically agree with the process or approach for animal research. And so often you don't have any clue that these facilities exist. They are not advertised. They are often very, very secure. I mean, bars on the windows, no sign on the door. So it makes it really fun for when an emergency responder has to find that building. It might just literally be a building number, like an address. You might not be able to get in easily. I mean, there is a lot of protections in place for security purposes.
So what kind of things would a first responder then expect as far as, I'm not going to say hardships, that's the wrong word, I'm trying to think of the right word, as far as things that would hinder their operation to, say, get to a medical emergency or some kind of an emergency inside the building? What roadblocks would they expect to find to be able to and have to overcome?
Well, that is a very open question. So first of all, finding the facility is going to be potentially challenging. I mean, it does depend on where you're located. It could just be a building in a suburban area. It could be a BSL-3 agricultural facility in the middle of a farm in the middle of nowhere. So we're getting into BSL-3 and 4. We didn't really touch on that yet, but we're talking high hazard pathogens. that have the ability to cause serious or potentially fatal disease that are often communicable by the airborne route. So you can inhale them. And they may not have treatment, prophylaxis, or vaccinations available. So that's why they up the containment into a higher containment level, which is BSL-3 or BSL-4, which is, I think, outbreak, spacesuits, right?
Like what?
Very few BSL-4s in the country, and they're very secret because many of them contain select agents, which we haven't got onto either. So there's another fun thing that you could encounter.
So there's not going to be a big neon sign saying, "Hey, this." No
neon sign.
Okay.
Finding the spot important, and this is where I would encourage first responders to engage with those stakeholders that have those types of facilities, and they have to declare them in permits.
Okay.
So your local county hazmat authority will have knowledge of what hazardous materials are in there. Your fire department, when they do fire department inspections of these facilities, because these facilities often have compressed gases, flammable solvents, because they're using them for research and also decontamination. They have large amounts of corrosive because they use them for cage washers. So your fire department, your local inspectors should have relationships with these companies already. Right. And if you can establish that relationship in advance, you know where the building is, right? So hopefully you can tour it in advance, kind of get a feel for, in a normal state, how do you get there? How do you get in? That's hurdle 2, right? So there'll be a lot of security requirements. You can, in theory, just take an axe to the door, but, worst case scenario, they often have Knox boxes, right? So it's establishing how to get in the building. And then when you get in the building, I guess we can then touch on what's in there, right? So we can talk about all the different potential hazards that you have in these live organism facilities. You could have all kinds of organisms.
I'm thinking optimally, if you had me respond to let's say a BSL-3, which, something comes in, I would hopefully be able to get maybe up to the front door or maybe just inside the front door and be like, bring me the problem because I'm not going any further.
Well, here comes the issue, right? So the problem could be that person is having a medical emergency inside the BSL-3 and they are covered with, they've fallen and they've covered themselves in nasty, right? So having a relationship with that facility, they will have an emergency response plan. They will have an internal procedure that hopefully is well drilled, everyone's trained on, and it's very unlikely that they will be lone working in that space because of the risk. So at this point, you're going to hopefully rely on the personnel working in the lab to remove and decon the person as much as possible and drag them likely into a gowning room. So most containment levels, 3 and 4 labs, I would say almost all 3 and 4, and some level 2, have what's called a gowning room. And it's kind of a buffer zone between the cold zone corridor and the hot zone lab. So this is kind of a staging area where they degown, gown in, and if they can get that victim out of the 3-level lab or 4-level lab into that gown area in a fully decon state or partially decon state, that would be a decision on the emergency responders' part if they're willing to enter that gowning area. So it's about getting information. They will have a biological safety officer. They probably have a high containment officer. They will be part of that emergency response plan, and they will likely be the point person for the emergency responder to connect with, to understand the level of risk that they're going into and what has been done to that point to get the victim to you versus going in.
Okay, so who is all right? So in New York City, we have we respond to a lot of utilities, and as a result, standard operating procedures are we go to the facility and we don't enter until what's known as a white hat comes in. So we know to ask and wait for a white hat, which is the person who knows the most about the facility and the situation. They're like the either the senior officer or senior person, somebody with a tremendous amount of knowledge. If I was walking into one of these labs or walking up to a facility, who would I say I would like to speak with this person, right? Because I don't just want to talk to somebody who works there, because the person that works there could be, the janitor, right? Which probably knows more about the facilities than anybody else. Not trying to put down janitors, but I want to know that the person standing there isn't like, Joe that was hired 6 months ago and he happens to be like the only guy working that day besides the person who went down. Like, and what questions should we be asking? Because right now we're in a position where we don't know that we don't know what we don't know. So it's really hard to gather information when you don't know that you don't know what you don't know. So if you were responding in, I know it sounds crazy, but it makes sense if you say it slowly. So if you were responding in as a first responder, like let's say you were headed, somebody was like, hey, there's a medical emergency in so-and-so level 3, level 4 lab, and you were riding along with them. What questions would you be asking as you were walking in the door and as you were about to, say, walk into the donning and doffing room, what we would refer to as the warm zone? Like, what are those sets of questions that you should be like, hey, buddy, you should ask this guy this question?
Okay. So just to, go back a second. Actually, most companies with those level labs do train their janitors because there's a risk to them. So the janitor might actually know more than you realize.
That's good point.
Valid. Again, also, not trying to put down, the janitorial
they're
arts.
Very valuable.
Yes.
Also, it's very likely anyone working in that facility will have knowledge of what's going on because they're highly trained to be present in that facility. So even if there is no one else than 6 months lab worker there, you'll be able to get information than them. But ideal case, what do you do if everything's in place? There are a beautiful emergency response plan internally. What do you do? Well, first of all, you want to ask for the biological safety officer. That is a person that is appointed by that company to be the expert in that facility to handle all things related to biosafety. So they will drive policy, they will enforce policy, they will create policy. They're responsible for the overall safety of that laboratory. It may or may not be a scientist that works in there. It could be an EHS person. It could be some other person who's been given that, but usually they have a shared background in safety and in biologics. So I would ask to have the biosafety officer present or available to communicate with.
Okay.
Then when you get there, you need to find out, one, what's kind of happened in the lab. Now, is that person having a medical emergency? And 2, are they contaminated?
Okay.
Right. So if they're just having a medical emergency and there's no contamination on that person, the risk to you is very low.
Yeah.
And then you can ask, is all the other agents contained? Are they all in storage? Are they all in the biological safety cabinet? There's no release situation, no release scenario, in which case it's perfectly safe for you to enter the hot zone potentially.
Would you? I would additionally say, hey, what I'm wearing right now? Am I safe with what I'm wearing? Do I need to upgrade N95 mask, whatever the hell it is? And the guy goes,
honestly,
yeah,
it would never hurt to put an N95 on because that is the level of respiratory protection that we use for biological exposure potential only. Sometimes they work with chemicals too, and they require different respirators, but if there is just a biological hazard that is respiratory, then N95 is your go-to standard respiratory protection for worker working in a biosafety level 3 or maybe 4. They might have more of a PAPA type respirator. But if you're entering any biosafety level 2 or 3 and there is no release potential, putting on an N95 is a very sensible option.
I like it. Like it. We have a plan.
If there's contamination, it gets interesting.
Okay.
So, first of all, part of the emergency response plan for a high containment lab, they will have supplies inside the lab staged that if they need to grab them, they therefore don't need to run around the building, peek out. Many BSL-3s have an AED in their facility so that they can just grab it. Many of them have advanced communication systems, walkie-talkies, at least a phone line, a hard phone line, so you can communicate with them. Many of them will have first aid supplies, or even things like, you remember those boards that the mechanics slide under the vehicles, mechanic dollies?
Ooh.
A lot of BSL-3s have those in there so that if a victim has gone down, you can roll them on it and dolly them out of the lab.
That's
to
brilliant.
Get them out of the facility.
That really is.
That's a great.
Why? How come none
It's
of our
great.
skids had wheels on them? Where is this?
What? That's a great idea.
They work great. I mean, they are the best thing I've seen for extracting a victim from any form of containment lab, be biological or chemical, API, active pharmaceutical ingredient, anything where there's a hazard, and you can roll them onto the mechanic's dolly and pull them out. Great.
Oh my God, I like that.
Yeah.
That's
so
actually a good one. I might have to steal that.
Yeah, please do. It works great. And then one thing we'll come to later is when you have an established relationship with these facilities, they love you drilling with them. So maybe you can go in and do a drill and play with that mechanic's dolly when you are doing a co-drill, the emergency response team internally, and you can actually get to play with those. So that's great.
See, I always imagine them being so super secretive. You know,
Yeah,
like you'd go knock on the door and a little brick comes out from the wall and
like,
there's 2 eyes and they're like, there's
what's the
nobody
password?
here, and it slides back in.
Most high containment facilities know that there is a risk for their employees, and if something goes wrong, they have a duty and they want to ensure that that person has the best chance of getting care as quickly as possible. And many have an established relationship with their local fire department and their local hazmat authorities, and they drill with them.
In a level 4 lab, and I know we're almost out of time. I just got this one question because I know this is going to be, within the first responder world, there are a lot of people that are not conspiracy orientated, but kind of untrusting of government, for lack of better words. And we do have disasters in the past where things have been massively downplayed. What in a large or a level 4 lab or even a level 3 lab, if there was some kind of a release, how upfront do you think that laboratory management is going to be to the first responders coming up, being like, yeah, no, there's some really bad stuff that just got out?
Okay, so for level 4s, there are very few in the country, and they're usually in government or military, CDC, NIH. There are a few in some academic institutions, but those labs are designed so nothing will escape.
Okay.
And it would have to be a catastrophic failure for those to actually release from the organizations. The facility itself is designed to contain it.
Okay.
So the likelihood of a release from those, unless it's like a tornado or, something where the entire building is completely obliterated. And even then, They're stored within safes, in freezers, in containers, and the chance of a release, and you're usually talking very tiny quantities, is very small. Now, also, these are highly government regulated. These are select agent labs. They have agents for bioterrorism in them, and the governments will be very highly involved in permits, inspections, and if something goes wrong in one of those facilities, they will be notified immediately, and they will be supporting the local emergency responders with that specific scenario. Now, that's 4s. For 3s, there are a lot more than you realize.
Numbers around the country?
Yeah, they are a lot. I mean, I want to say there's probably at least 25 in San Diego. Just in San Diego.
Whoa!
At least. There may be more than that. So, and you don't know about it.
I'm going to tell Todd.
They are not advertised because of the nature of their research.
Sure.
So for BSL-3s, they also may have select agents in them. They may not. But they will also have very stringent permits and guidelines on how they are permitted to operate to promote containment, including in emergency situations. So the CDC or USDA, if it depends on if it's an agent that is impactful to human health or animal health and the environment, you have 2 governing bodies. They will only issue a permit for use of those materials if you have the facility approved, the emergency plans in place. And again, these are containment facilities, so it is unlikely they will be breached. Now, your question came back to, well, what happens when, is an emergency responder going to kind of freak out? And there are hazards in there. It's a genuine risk, right? Yeah. So the most important thing, coming back to, finding the biosafety officer, finding out the nature of the emergency, and then finding out if there's contamination present. If there's contamination present, that's when it gets more challenging. And then you have to find out the nature of that contamination. But these containment labs have emergency internal response teams that are trained to deal with this. And the goal is to get that victim out to you in a non-contaminated state so that you can then deal with the medical emergency.
I definitely want to hear more about all of that, but I do know that I 100% also know that there is a cab waiting to pick Bob up to go teach in California. So,
well, you know what? I know. And in the next episode, we're going to go through all 12 times they did release BSL-4s. I'm totally joking. That's a total joke.
It doesn't happen. You don't. It doesn't happen.
That's a cliffhanger. That's what they call a cliffhanger. They got to come back for it.
Oh no! Next time we're going to talk about the animal facilities and the insect facilities, because they present very unique hazards on top of just like, imagine a radioactive rabid llama running at you versus a little thing in a petri dish. Right?
I can. So, I heard that's where flying cockroaches were developed. They were actually developed in a lab.
Come on. All right. We'll see you guys next week.
Have a good one.